Professor Jha, Professor Joisna Meshram, my young friends sitting over here, research scientists and younger scientists who are there. So I shall discuss here some recent aspects for estimation of different types of, or I can say recognition, molecular recognition, different types of molecules by taking an example of antioxidants. I have taken here an example of eugenol, commonly known as long, how we can estimate and monitor the amount of antioxidants in a person while giving a dose of antioxidant, whether antioxidants are required or not, or by electrochemical method. I shall discuss here voltammetric method and development of voltammetric sensors for molecular recognition with the aim that a sensor can be developed so that like a pH strip, pH paper strip, it can be dipped in a solution and immediately we can find out the concentration of a molecule present there, whether it is an antioxidant, pharmaceutical or any other type of molecule, for which type I have developed a sensor which I am discussing here. How this is the general outlay of the lecture, how sensor is developed, sensor is fabricated by using different techniques, sensor is characterized and then how it is applied for qualitative and quantitative estimation in different matrices. It may be pure matrix or it may be in blood or it may be different types of pharmaceutical combinations or different types of matrices. It may be present there. So these nanocomposites have been intensely investigated due to the performance improvement achieved when a small amount of nanosized particles are added to a polymer matrix. By this incorporation of these polymer nanoparticles in a composite, different types of carbon particles such as carbon nanotube, graphene, nano oxide, bismuth oxide, different types of metal oxide, we can prepare nanocomposites and then apply them after characterization as sensors, and we can reach up to nano level and pico level. Even we can compare it with the ELISA test which is commonly available for that purpose. So these are the various properties that have been studied by many researchers, which include biocompatibility, abrasion stability, anti-static property, moisture management, UV protection, water, oil and soil repellency, flame retardancy, antimicrobial activity, biocatalysis, all this is being studied. Why we study nanocomposite? Why electrochemical analytical techniques? These days, there is a general feeling when electroanalytical techniques are such sensitive, then why they are not used in industry? Why industries, particularly pharmaceutical industries, are not substituting them? The reason is that because we do not have experts, well-studied interpreter of electrochemical data. Otherwise, these techniques are very inexpensive and require very simple management, and it can be applied without any sample preparation. There is no need to any lengthy analyte preparation. These techniques can be applied directly to there. These are the experimental setup, methodology, simple methodology, then characterization of fabricated sensor. Sensor characterization very important part. It should be very well characterized by different techniques. Well, whatever technique, it should be exhaustively characterized so that results are reproducible. In this case, they have been characterized by NMR, FTIR, SAM, TEM, impedance spectroscopy. They have been totally characterized, and after that, they have been applied as a sensor. This is the characterization of fabricated sensor by different techniques. Here is a Raman spectra, then it is the SAM, how they differ in the when the oxides and nanographenes are incorporated in the polyaniline nanocomposite. This is the characterization of fabricated sensor by electrochemical impedance spectroscopy. It clearly tells that how with the incorporation of graphene oxide and glassy carbon electrode, graphene oxide, glassy carbon electrode, how the conductivity is increasing and resistance is being decreased. So this is very helpful in characterization of the sensor, developed sensor. Then electrochemical characterization of fabricated sensor. These are the A, B, C, D, 4 cyclic voltammogram of different uh, uh, potassium ferrocyanide system. How it works at bare glassy carbon electrode, modified electrode B, further modified electrode C and modified electrode D. Clearly indicating that with the modification, current is increasing, whether in pure potassium ferrocyanide at glassy carbon, bare glassy carbon electrode, current is very small current. But with modification, the current of potassium ferrocyanide system is increasing, clearly indicating the increased sensitivity of the developed sensor. This is the characterization of again electrocatalytic effect of fabricated sensor by using different types of differential pulse voltammograms. Here we are taking, then we have taken anodic stripping voltammetry, cathodic stripping voltammetry, and different types of. Overall, we have applied 21 variations of electroanalytical technique for finding out the total mechanism. We have put the mechanism, we have isolated the end product by different techniques, and further quantification is a significant aspect of the study. This is the simple formula of antioxidant analyze which has been reported here, which I am discussing here. This is eugenol, commonly known as long. Long is a very important substance in Hindu mythology, is used in everywhere whenever a fire decision is there. It is long, low, significantly known as botanical name and possess excellent antifungal, antibacterial, anticonvulsant, antiviral, antioxidant, anti-inflammatory properties. Also works as potent platelet inhibitor to prevent blood clot, fragrance and flavoring agent. So as to maintain and to find out which concentration of eugenol is optimum, this study may help. Although I have no direct information about the eugenol requirement for the body, but such type of study can help in monitoring the important constituents which is required in the body. This is the result and discussion part. How optimization of experimental conditions. First, we have to find out the optimum pH. What should be the pH of the study? At what pH, optimum pH study should be taken out, and at what pH is current is maximum. Then calibration curve, linearity, detection, and quantification. This current is continuously increasing if we are increasing the concentration of eugenol. After quantifying the Pani graphene oxide glassy carbon sensor and characterizing it and characterizing how much amount of modifier is required for glassy carbon electrode and what should be the pH, we have done calibration studies and a state curve is there, and we can easily find out the concentration from this, and it goes up to this. This is reproducibility experiment. Naturally, when we do any analytical experiment, we find out the selectivity and sensitivity. So also reproducibility. So this is the reproducibility of the experiment. And then accuracy, precision and stability were also studied, and it was an excellent method developed for that purpose. Analytical applications, different types of analytical applications are there. Then similarly, I may here maybe somewhat different that these sensors can be applied for sensing of chemical warfare agents that we have used for finding out different types of chemical warfare agents. Similarly, we have studied it different types of finding out electrochemical immunosensing of staphylococcus enterotoxin B trap using voltammetric sensor. So we want to develop, our aim is, although so far we could not develop a strip of paper, a very sensitive, ultra sensitive uh, thin paper technique which can, on dipping this paper, a very sensitive sensor into blood or sample, we can find out the development of the disease, onset of the disease, we can find out. That is the aim of these electroanalytical studies. And further, we want that these techniques should supplement our existing HPLC techniques which are used in different pharmaceutical industries, which are very costly, require tedious sample preparation and time consuming. Where our technique hardly takes 1 minute and no sample preparation, we can directly sense the substance present there and we can quantify it immediately. 4 or 5 or 6 substances can be quantified directly by using this technique. So these are the different studies we have carried out for carrying out different types of microbes in the body. Different types of microbes we have studied here. And we have published all these work in RSC advances, sensors and actuators, biosensors and bioelectronics. So different types of these electrodes have been synthesized. And this is my research group. Research group of our 20 students are working with me. Some of them are here and they have accompanied me, and I'm thankful to them for working for me.
Thank you very much.